Jessica. And thank you so much for that introduction. I'm going to make sure that I can share these.
So we
Okay, great.
also have these poll questions that we can put up on the screen.
So this poll will help me as I decide how much time to spend on reviewing previously covered topics in this webinar series.
So we'll give it about 30 seconds.
And want to thank folks in advance for bearing with me today. I have a bit of a cold, but I'm at home, can't infect others. So we'll get through this today, and I'm looking forward to the Q&A and discussion afterwards.
Okay, so there's a pretty mixed group on the line so far today. So
Great.
that's fantastic.
And we have one more poll. Do you think that the environment has an impact on the effectiveness of potential cure strategies? And we'll discuss what we mean by environment as well in today's presentation.
Great. And for those who are just joining us, welcome. We are starting with just some poll questions to get us started before we dig into the content. So I think it's safe to say that everybody is here because they do think there is some impact on the environment, on the effectiveness.
Great. Well, we'll talk about this, and today, and in particular, I think you'll see a theme is that there's much left to be learned, much that we still don't know. So we'll discuss all of these topics today. But first, I wanted to start by providing a review of HIV Cure 101, which was covered in previous presentations in this series. But I see there are about half the folks here are new to this series. So we'll go through HIV Cure 101 relatively briefly. And I wanted to first start by talking about the HIV life cycle, which is important to understanding HIV biology and what we call the HIV reservoir. So this figure, this schematic, shows a detailed version of the viral life cycle. It's very detailed. It's not important to cover all of the details for today's talk. However, I did want to cover the main steps of the life cycle. So when an HIV virus directly infects a cell, it first attaches to that cell using a receptor on the cell surface called CD4, as well as a co-receptor. And this co-receptor can be CCR5 or another co-receptor called CXCR4. And HIV uses these cell surface proteins to attach to the cell. This process can actually be inhibited by classes of drugs called fusion inhibitors or CCR5 inhibitors. These are classes of antiretroviral drugs, and they're shown here in these red lines. So after HIV attaches to the cell that it's infecting, it then fuses with the cell, undergoes uncoating, and releases the contents of the virus into the cell. So this includes the genetic material, HIV's genetic material, as well as proteins, enzymes that are necessary for the rest of the viral life cycle. So HIV is a retrovirus, which means it's an RNA virus, and it undergoes a process called reverse transcription, whereby its RNA genome is converted into DNA by the help of an enzyme called reverse transcriptase. And then the DNA, the new viral DNA, is imported into the nucleus of the cell that it's infected. And this is a very important process here called integration. And so the viral DNA, with the help of an enzyme called HIV integrase, is integrated into the DNA of the cell that is infecting. So the new viral genetic material is permanently integrated into the genetic material of the cell that it is infecting. And once this happens, the cell then is permanently infected with HIV. And so this is a really important part or step of the viral life cycle. We call this integrated HIV genome a provirus. And so then I wanted to highlight these steps shown called transcription and translation. So in active viral replication, where new HIV virions are being produced, HIV is being transcribed, so viral RNA is being transcribed or being made from the new viral DNA genome. And then this viral RNA is translated into proteins, and new viruses are assembled and bud off of the infected cell. However, sometimes a cell is what we call transcriptionally latent. So that means that no RNA is being produced from the HIV DNA. So this process of transcription is not happening. However, the HIV provirus is still there. It's still in the infected cell. And we call these cells latently infected cells or dormant cells. And these latently infected cells are an important component of the HIV reservoir. So what is the HIV reservoir? Well, a narrow definition of the reservoir includes all infected cells in the body with genetically intact proviruses that are capable of causing rebound viremia if antiretroviral therapy is interrupted. And a more broader definition of the reservoir, which you may also see used, it's important how we define the reservoir, includes all infected cells regardless of the genomic or genetic intactness of that provirus, as well as its transcriptional activity. So as I mentioned, this traditional concept of latently infected cells states that no HIV RNA is being produced from the integrated provirus. However, there is some emerging evidence to suggest that latency may be a continuum ranging from little or no viral transcription through to transcription. So actually, in most people living with HIV, we can detect HIV-infected cells that are actively transcribing HIV through to translation, and even in some cases, new active virion production. So sometimes, in some people, infected cells may actually go on to produce virus despite antiretroviral therapy. However, there's not evidence for ongoing viral replication under antiretroviral therapy in the absence of drug resistance. So these infected cells that may produce new virus may be persisting for other reasons, and we're going to discuss that today. And then also there's some evidence that defective proviruses, these are genetically defective proviruses that cannot produce new whole HIV viruses, may be capable of producing HIV transcripts as well as proteins and may be clinically relevant, and more research is needed in this area. So CD4 T cells, you may have heard of these cells. They're very important immune cells that are the primary targets of HIV infection. They are also the primary reservoir harboring cells. And other cell types in the body may also be reservoir harboring, such as microglia, macrophages, astrocytes. I'm going to discuss this on the next slide. But the significance of these other cellular reservoirs is still being studied. So to further illustrate the HIV reservoir and its complexity, I thought this was a great figure from the most recent International AIDS Society Global Scientific Strategy for an HIV Cure. And this figure shows how the HIV reservoir can be defined across a number of dimensions, including shown in this outer ring, the anatomic and microanatomical location of the reservoir. So the gut is a very important tissue site for the reservoir. Also, lymph nodes are important. So lymph nodes may exclude, they may be immune privileged sites that exclude immune effectors that can eliminate HIV-infected cells. And so there may be sanctuary sites in lymph nodes where HIV-infected cells can hide out. And there may be other important anatomic compartments, spleen, liver, the central nervous system. So not just peripheral blood. This next ring shows the cell type. And so these lymphocytes, these are the CD4 T cells, the immune cells that I was describing that are the primary targets of HIV infection. But other cell types may be important reservoir harboring cells such as macrophages. Astrocytes in the brain may also be important. These play a role in supporting neurons, and there's a lot of these cells in the brain. So the cell type is also important when considering the reservoir. This next ring shows the cell properties or the cell functional profile. And an important functional property is whether the cell is in an activated or resting state, particularly for CD4 T cells, where activated cells may be more likely to produce HIV, and resting cells may not have the necessary factors to produce new HIV viruses. Also, this concept shown here, resistance to immune killing, this concept has really gained traction in the past few years. Our lab and other labs have contributed research to this question on whether HIV reservoir harboring cells may be resistant to killing by the immune system and may be selected for over time for resistance to immune-mediated killing. So this is very much an active area of research. And then, as I also mentioned, the transcriptional activity of the cell is an important functional property. And then lastly, this inner ring describes the provirus sequence and location. So where exactly in the human genome has the HIV DNA integrated? And we call this the integration site, the genomic integration site. And we think this integration site may be important for HIV biology and HIV persistence, but also may be relevant to HIV cure strategies, certain HIV cure strategies. And then, as I mentioned, whether the HIV provirus is genetically intact or whether it is genetically defective. So there are defects that mean that this HIV provirus cannot produce new viruses. And this is important because in most people living with HIV, only about 2 to 5% of HIV proviruses are genetically intact, and the remainder, 95 to 98%, are genetically defective. Okay, so I made this figure to illustrate the clinical relevance of the HIV reservoir, which is that it serves as a source of viral rebound upon therapy interruption. Here, when someone first acquires HIV, I've shown virus levels in blood in red. And so virus levels in the blood increase exponentially to what we call set point viremia, and then gradually decrease to and plateau. Sorry, this is peak viremia. Then viral levels gradually decrease and plateau to what we call set point viremia, following the development of an adaptive immune response that partially controls viral replication. And then CD4 cell counts are shown in this blue line. These initially decline, but then recover following partial immune control of viral replication. And then when someone starts antiretroviral therapy, this stops the virus from replicating and allows the virus to drop to undetectable levels in the blood. And then CD4 cell counts during the course of antiretroviral therapy continue to recover, and other immune health continues to improve. However, if therapy is interrupted, the virus can begin replicating again from the reservoir. So I've shown the HIV reservoir here in this brown line, and it is established very early in the course of HIV infection, and it also persists throughout therapy. And so when antiretroviral therapy is interrupted, the virus can begin replicating again from these reservoir harboring cells and can rebound in blood, which I've shown here in this red line, to detectable levels, usually within weeks in most people living with HIV. There are rare individuals called post-treatment controllers. Happy to talk about that. I'm not going over that today, but happy to talk about so-called elite control, post-treatment control during the Q&A. So measuring the HIV reservoir is important. I think, this is a quote from the IAS strategy for a cure, that significant progress towards a cure for HIV depends on having sensitive, specific, and quantitative measures of persistent virus that can be applied to various anatomical compartments. So there are multiple measures to quantify different aspects of HIV persistence in the reservoir. I'm only going to go through a few today, but these are perhaps, I think, important ones relevant to today's talk. The first is cell-associated HIV DNA. So this measures total HIV DNA levels, including all intact as well as defective proviruses together. So this assay measures all HIV genetic material. Then cell-associated HIV RNA. This measures levels of various HIV RNA forms, i.e. HIV transcription, this process of transcription I noted earlier. We can also measure plasma HIV RNA or cell-free HIV RNA in other fluids such as cerebrospinal fluid. And this measures the amount of cell-free virus in that fluid, such as blood. And this may represent HIV virus production. There is an assay called the quantitative viral outgrowth assay. This measures what we call inducible replication competent virus levels. So here we take cells from blood from a person living with HIV. We put them in a cell culture dish. We stimulate those cells with different agents, what we call ex vivo, and we try to see how much HIV grows out of those cells. And so this measure provides a definitive minimum estimate of the rebound competent reservoir size. And then a newer assay is called the intact proviral DNA assay. This is a sequence-based assay which separately quantifies genetically intact and defective HIV DNA levels. And this assay provides a definitive maximum estimate by intact DNA size of the reservoir size. So I also wanted to briefly touch on immune responses to HIV, as these may be harnessed in HIV cure strategies. And broadly speaking, immune responses can be divided into innate immune responses and adaptive immune responses. So innate immune responses, these are very quick to develop, but are also not specific to the virus. So here, pattern recognition receptors on cells recognize conserved molecular patterns on pathogens such as HIV. And this triggers host responses in that cell, including the induction of inflammatory cytokines, as well as production of antiviral interferons, which then recruit and activate cells of the innate immune system, innate immune cells. These include macrophages, cells called natural killer cells, as well as dendritic cells. These are antigen-presenting cells I'll describe in a moment. And this initiates the development of adaptive immunity. So adaptive immune responses to HIV take longer to develop, but also persist for longer, up to many years, and are also HIV specific. And so you may hear about harnessing these adaptive immune responses in the context of certain immune-based HIV cure strategies. So here, upon viral infection, specialized cells called antigen-presenting cells. Here I'm showing an important cell type called dendritic cells. These are professional antigen-presented cells. They take up parts of the HIV virus and present them to cells called T cells and B cells, which are the two major cell types of adaptive immunity. And this primes these cells for development and the specific ability to eliminate HIV-infected cells. So I also want to touch on language for HIV cure and what we mean when we say HIV cure. So these definitions come from the community preferred language document developed by AVAC and the Martin Delaney Collaboratories. So when we talk about defining HIV cure, traditionally, this classic concept of cure, this classic definition of cure, has been taken to mean removal of all replication competent virus from the body of the person living with HIV. And some preferred alternative phrases may include classic cure or just cure or eradication. In some cases, in some very rare individuals, maybe spontaneous cure as well as traditional cure. And so this term can be used to describe individuals who have received stem cell transplant from a donor who has natural immunity to HIV. There have been at least 5 individuals who have probably been cured of HIV through this bone marrow stem cell transplant method. Or it may also refer to an individual who has cleared the virus, through immune-mediated mechanisms in the absence of antiretroviral therapy, such as Lorene Willenberg or the Esperanza patient. So this is the classic concept of cure, but another concept in cure is viral control. And so the definition here is immunologic suppression of the virus or some other form of suppression of the virus to undetectable levels off of antiretroviral therapy. So some preferred phrases to this are durable control, viral control off of therapy, immune-based control, or antiretroviral-free virologic control. And this may describe a person with HIV-specific immune markers and maybe a detectable inducible reservoir, such as by this QVOA assay that I mentioned, but are still able to remain undetectable or at least very low levels of virus in blood off of therapy. And this is important because viral control is probably or likely a more realistic and more achievable goal in HIV cure. So I next want to touch on some cure strategies under development. And this is not going to be a comprehensive list. And I'm not going to go into the details of these specific strategies, but I'm happy to discuss them further during the Q&A. So the first include immune-based strategies. So this can include shock and kill, or what's also called by folks, I think on the West Coast, call it kick and kill, and a related concept of reduce and control. So this proposes to use drugs called latency reversing agents coupled with other immunotherapies to awaken the latent or dormant HIV and then to eliminate those HIV infected cells. And this related concept proposes perhaps we don't need to eliminate all HIV infected cells, just reduce the number to very low levels, which then maybe can allow for immune mediated control long-term off of therapy. Broadly neutralizing antibodies. These are, I think, really vaulted to a promising approach to HIV cure and are being tested in multiple clinical trials right now. T-cell based therapies such as chimeric antigen receptor T cells and other engineered T cells, either engineered ex vivo or in vivo. Therapeutic vaccination, which means a vaccine given to someone living with HIV to improve their immune response to HIV, which could possibly promote control of the virus off of therapy. And then also immunomodulatory drugs are being explored. These can include drugs like IL-15 superagonist, immune checkpoint inhibitors, and other immunomodulatory agents. Gene editing strategies are also being developed and explored. This can include strategies called block and lock. So the concept here is to block and prevent the HIV provirus, to lock it in a permanently transcriptionally silent state and prevent it from ever being able to produce new viruses. So if we could do that for all HIV infected cells, then no new viruses could be produced. It's a strategy that's exploratory. It's in the very much in the basic science research stages right now, but that's the concept. Also, viral excision or permanent silencing by CRISPR-Cas9 gene engineering is being explored. And then also engineering this mutation in the CCR5 co-receptor called the Delta 32 mutation, which is the mutation in the cases of stem cell transplant in the donor cells that confers natural resistance to some strains of HIV and may have allowed for those individuals to achieve HIV cure. And then, it's likely that combination approaches, some combination of all of these approaches may be needed to achieve cure. And it's not likely that one approach will be applicable to every person living with HIV. And we're going to discuss that a little bit later when we talk about environment and how environment may impact HIV cure approaches. So I'm not going to go through this figure. It just comes from a review that I co-authored on immunological approaches to HIV cure, showing ideas for next generation combination HIV cure trials, some of which clinical trials are occurring now, which is exciting, and we'll see what these clinical trials show. So Jessica Prodger, Dr. Jessica Prodger in the previous webinar covered sex and gender and HIV cure. So I'm only going to briefly touch on this, but it has been reported that biological sex as assigned at birth can influence HIV pathogenesis, immune responses to HIV, the person's response to antiviral therapy, how effective antiviral therapy is, HIV reservoirs, which may be less transcriptionally active in women. This was from one study conducted in San Francisco that found women may have lower transcriptional HIV activity. May not be true for all women in all parts of the world, but this was just from one study. And then one study in Rakai, Uganda found that HIV reservoirs may be less inducible in women. And these differences by biological sex can be driven by multiple factors, including the influence of sex hormones such as estrogen, sex-specific epigenetic profiles, genetic differences also derived from chromosomal complement, as well as, importantly, behavioral and socioeconomic dynamics that may differ by sex. So, some important concepts in sex and HIV cure. And it's also maybe true that not the cure strategies may also need to be tailored to be sex specific. And so we'll talk about that later as well. So some additional concepts in HIV cure that I wanted to cover. I think when we talk about HIV cure and someone's location and their environment, it's important to also consider these concepts when developing a potentially curative intervention strategy. So the first concept is safety. Is the intervention safe with few or no serious adverse events in most people living with HIV? Next is efficacy. So is the intervention effective? And so does it, one, maintain viral loads below the transmission level, which has conservatively been set at less than 200 copies per milliliter, or at least at some low level? And two, does it promote the health of the person living with HIV off of therapy, at least in a proportion of people living with HIV? So there was this publication a couple of years ago now on a multi-stakeholder consensus on a target product profile for an HIV cure, which used surveys and working groups to try to define the minimum and the optimal characteristics of an HIV cure intervention. So in this publication, in this target product profile, they identified that the minimal accepted efficacy would be around 20%, meaning the intervention works in at least 20% of people living with HIV, and the optimal efficacy would be in at least in over 90% of people living with HIV. That would be optimal for a cure strategy. Durability. So does the intervention induce long-term control without the need for frequent or for any subsequent interventions? And so the minimal acceptable criteria from this target product profile was once per year, so an intervention given once per year. And the optimal intervention would be an intervention that's only given once, and then you never need another intervention. And then scalability. So this is related to cost and cost effectiveness. Is the intervention cost effective, and can it be scaled up and produced to be given to the entire population of people living with HIV that it may be effective in? So scalability is also, I think, a really important concept when we're talking about cure strategies going into development. So we know that antiretroviral therapy is life-saving. However, these statistics are from the most recent UNAIDS fact sheet. In 2022, 39 million people globally were living with HIV, but only 76%, so 29.8 million people, were estimated to be accessing antiretroviral therapy. So that means that about 9.2 million people globally did not have access to therapy in 2022. And it was estimated that only 57% of children ages 0 to 14 years were estimated to have access to therapy. So there are people still globally that don't have access to antiretroviral therapy. Also, sub-Saharan Africa accounted for 50% of global population in 2022, but 68% of people living with HIV and 57% of new infections. So when we're talking about an HIV cure, I think it's also important, and particularly in comparing it to antiretroviral therapy, it's an HIV cure could reach and help people living with HIV who cannot access or adhere to antiretroviral therapy. It could also improve the individual's quality of life by reducing comorbidities, treatment burden, stigma associated with living with HIV, as well as socioeconomic burdens. And it may also present a financially sustainable solution to ending the epidemic. So with that, we'll take our first Q&A break, and we'll take some time here for questions and discussion.
This is absolutely fantastic. You have a number of questions that have come in. I do see Joyce. I do want to get your voice into the fold. So Joyce,
We
can
have a
you?
lot of expertise on this call, so really glad
Joyce,
to.
can you just unmute yourself? You should be unmuted. So Joyce, you should be able to unmute. Okay, well, while we wait for Joyce to ask her question, a few came in on the chat. So first of all, if a cell is generating HIV virus, how can it still be classified as latent? And can you just say what qualities are included in latency that distinguish it from cells actively replicating virus?
Yeah, as I mentioned, a strict definition of latency is transcriptional silence. That's what we mean by transcriptional latency. But I think an emerging body of evidence makes it clear that not all HIV infected cells are transcriptionally latent. There may be other blocks to producing viruses. There may be blocks to translation, for instance. So there may be other blocks besides transcriptional blocks that prevent a cell from producing new viruses. So this in and of itself could be considered a form of latency. But it's important to recognize that in the biology of the HIV reservoir, that there are cells that are actively transcribing HIV and even actively producing proteins. These cells may be visible to the immune system, whether the protein is coming from genetically intact proviruses or defective proviruses. There's still much research that's needed on this area, but these cells may still be visible to the immune system. They may potentially distract immune effectors. They may evolve by coming in contact with the immune system. They may potentially evolve immune resistance over time. So cells that harbor properties that make them resistant to killing by immune effectors may be selected for over time on therapy for immune resistance. And this is a really important concept that is gaining traction in the field. Can you repeat the second part of the question, Jessica?
So the second part was what qualities are included in latency that distinguish it from cells actively replicating virus?
Yeah, I think, it depends on how we define latency again, whether we think of it as a continuum. These measurements are how we look at latency, whether cell-associated RNA, and there are different forms of cell-associated RNA. So the primary HIV transcript is called unspliced, but HIV also goes multiple splicing into different isoforms, and we can look at these different splice forms. So these measurements of HIV RNA, we can also measure HIV proteins being produced, like P24, for instance, by sensitive assays. And so these measurements are how we look at the various stages of the life cycle and whether or not cells are latent. Hopefully that answers the
So
question.
yeah, I think perhaps a good way to think about it is just that there's a lot of information that is continuing to be discovered and uncovered with, as the science advances. And what I think what Dr. Ward is saying is that there's this growing body of evidence that says it's not just about the fact that HIV can't transcribe or come out or form new viruses or begin to replicate, but that it could be active, but yeah, that there's these other
Maybe
blocks, and that there's this body of
Yeah, and
evidence that
maybe
we need to think about.
an important example of that is in some people living with HIV, despite effective therapy preventing new rounds of viral replication, they may have detectable viral loads either by clinical assay or by sensitive, what we call single copy assay. And this has concerned clinicians for a long time, even in ART intensification studies. So when new antiretroviral or additional antiretroviral therapies were added to someone's existing ART regimen, this did not eliminate the detectable viremia. And there's been really interesting recent findings in this regard that some infected cells may be able to produce virus. So their HIV transcription is happening, new virions are being produced by this infected cell, but these cells for some reason are not, and it's usually clonal population of cells, but for some reason these cells are not eliminated by the immune system. There's been some also recent findings from Francesco Simonetti's group, Sabrina Brume and others, that in some cases, defective proviruses with defects in specific regions like the 5' leader sequence may be able to produce viruses that are detectable by clinical viral load assay, but aren't actually replication competent viruses. And these cells that are producing these defective viruses may also persist on therapy. So that I think is an important example. Not everyone has detectable viremia on therapy, but some people do, and it may not be because of antiretroviral therapy failure or development of drug resistance.
Hmm. So one more question, then I want to get into the environment elements. So one is just on this idea of measurement is when would each method to measuring the reservoir be used? And would determining the amount of replication competent virus be the important measurement for cure?
Yeah, so for cure trials, the really the gold standard to see if a curative intervention has an impact on the reservoir is analytic treatment interruption. And so here a person is asked to stop their antiretroviral therapy, and we look at how long it takes for someone to rebound, as well as the kinetics of rebound. So what that curve looks like, if it's exponential or if it's not, what that curve looks like following viral rebound. So that's the gold standard in cure-related clinical trials. Excuse me, but we also do look at measures of the reservoir, particularly in curative interventions. We look at IPDA is being used, and then some trials are also using QVOA to look at changes in the size of the replication competent viral reservoir, and by IPDA to look at changes in size of the intact HIV DNA reservoir. So those two measures are important in curative intervention trials, but ATI, analytic treatment interruption, is the gold standard to see if a curative intervention has an impact.
Great. And so I see there's a number of questions that have come in. I think some of them we'll get into with the environment. And so
Yes,
I do
great.
want us, yeah, to kind of move through, and then we'll be able to get to the next set of question blocks.
Great, wonderful. Okay, so we'll move on. There we go. So next, going to move to the topic for today's talk on environment. So location, clade, co-infections and comorbidities, and ART access. And I think it's important to note that these may all be related concepts. So a major challenge with HIV is its formidable genetic diversity. So there are two major types of HIV, HIV-1 and HIV-2. But HIV-1 is responsible for over 95% of all infections globally. And HIV-1 is divided into 4 different groups, M, N, O, and P. And group M is the most widespread worldwide. This is the group responsible for the global pandemic. However, within this group, there's also a lot of genetic diversity, and it's further subdivided into what we call subtypes. So you may have heard about HIV subtype, and group M HIV-1 is divided into 9 distinct subtypes. Those include A, B, C, D. There are other subtypes. I think it's important to mention that subtype C dominates the global epidemic. So that's shown in this pie chart here on the right. These data are a few years old. They come from a publication from 2018. But nevertheless, subtype C accounts for over 50% of people living with HIV globally. But subtype B, which is shaded here in brown, is the most prevalent subtype in the Americas, North America and South America, as well as in Europe and Australia. And then I also wanted to mention that 2 or more subtypes can combine to form a hybrid known as a circulating recombinant form. So you may hear about these CRFs also. So this figure on the top, this depicts a phylogenetic tree. This is a tree, a visual way to show genetic relatedness. And this shows the distinct genetic lineages of HIV-1, all here within group M compared to other groups, along with relationships to simian immunodeficiency viruses. So HIV-1 is most closely related to SIV viruses circulating in chimpanzees and gorillas, whereas HIV-2 is most closely related to SIV found in Sudi Mangabe. And then this figure 2 shows the global distribution of HIV subtypes from 2010 to 2015. So again, note that subtype C is the most prevalent subtype globally, where subtype B virus is the major subtype found in North America, Europe, and Australia. So this figure, it's a little bit older now, but it shows the genetic diversity of HIV here compared. So this shows the total genetic diversity in this phylogenetic tree of the global influenza outbreak in 1996. And then this phylogenetic tree shows the total genetic diversity of HIV in a single person living with HIV for 6 years. This tree shows HIV genetic diversity in a cohort of people from Amsterdam living with HIV in 1991. And then this tree shows the genetic diversity of HIV circulating strains found in Congo region in 1997. So you can really appreciate this immense genetic diversity of HIV. This is because HIV, when we're talking about the biology, it has, when it's undergoing reverse transcription, it has an error-prone reverse transcriptase enzyme, which makes mistakes, which may sometimes be, usually are deleterious to the virus, but it sometimes may confer some advantages to the virus so that it can mutate around antiretroviral therapy pressure, around immune pressures, and this leads to a lot of genetic diversity. So a challenge in HIV Cure is that most persistence and cure studies to date have been conducted on populations living with subtype B HIV. So there have been few comparative studies on subtype B versus non-B. We also need to adapt some reservoir assays, particularly sequence-based assays, to non-subtype B HIV. We also need to build capacity for basic research and clinical trials in high-burden, resource-limited settings, which are primarily impacted by non-subtype B HIV. And then also there's a important question: what is the role of viral subtype and maybe relatedly geography and ethnicity on the effectiveness of cure interventions? So these, this is really unknown. The answers are unknown as of now. HIV Cure clinical trials are in very early stages, phase 1 and early phase 2 trials, and may not include large numbers of participants of diverse viral subtypes. So I mentioned there are a few comparative studies between B versus non-B. I'm only going to highlight 2 here that did explicitly compare subtype B to a non-subtype B HIV. And so this first study compared what we call IUPM, infectious units per million. This is the readout of the replication competent reservoir by this QVOA assay. This was measured in a cohort of people living with HIV in Rakai, Uganda, who were all living with either subtype A or subtype D HIV, versus a cohort of people living with HIV in Baltimore, Maryland. And these individuals were all living with subtype B HIV. And very interestingly, in this study, they found that Ugandan participants had a 3-fold lower replication-competent reservoir size by QVOA versus the Baltimorean participants. And they found that this difference in the replication-competent reservoir size persisted despite controlling for differences in multiple relevant clinical factors. So this is a statistical procedure whereby you take into account other important clinical factors such as CD4 count, pre-ART viral load, maybe other important factors relevant to the size of the HIV reservoir. You try to statistically control for those factors. And even after controlling for those, there was still this difference in reservoir size in Ugandans. And so the biological mechanism here is really unknown. One limitation to this study was that it was cross-sectional in nature. It wasn't longitudinal. So this prevented the researchers from knowing whether the diminished reservoir size in Ugandans was due to a diminished reservoir formation early in the course of therapy or at the timing of antiretroviral therapy initiation, or whether it was due to an accelerated decay in the reservoir over time on therapy. Also, they noted that subtype D, which I believe around 60% of these participants in Rakai were living with, is most genetically similar to subtype B with similar viral dynamics, such as CXCR4 usage. So maybe this difference is not driven by viral subtype, but we do not know. Maybe there are other environmental factors driving this difference. This study compared inducible reservoir size by an assay called the EDITS assay, and they also compared total HIV cell-associated DNA in a cohort of people living with HIV from Kampala, Uganda, who were all living with either subtype A, D, or C, versus a cohort of people living with HIV from Cleveland, Ohio. These participants were all living with subtype B HIV. And they found that the Uganda participants also had a lower inducible reservoir, excuse me, as well as lower total HIV DNA levels. However, Uganda participants had greater reservoir genetic diversity compared to the participants from Cleveland. So in their discussion, they noted that the smaller but more diverse peripheral HIV reservoir in Ugandan participants might be associated with viral factors such as non-B subtype, or it could also be associated with host factors, such as a higher incidence of co-infections and comorbidities, leading to a loss of clonal expansion. So this research, just these two studies, taken together, I think really highlight the need to understand reservoir dynamics in diverse populations as part of ongoing efforts to find a cure for HIV, particularly in low and middle income countries. So I also mentioned the need to adapt some reservoir assays to non-subtype B HIV. There has been some recent progress on this front for sequence-based assays. So our group and others have adapted the IPDA, the intact proviral DNA assay, to be applied to other subtypes, such as subtypes A, D, C, and then this cross subtype IPDA was also developed. So there has been some encouraging progress on this front and measuring other subtypes accurately. I also mentioned the need to build capacity for basic research and clinical trials in high burden, resource limited settings, which are again primarily impacted by non-subtype B HIV. So I wanted to briefly highlight this HIV Cure Africa Acceleration Partnership as one model to build capacity in a high burden area. I'm not going to go through the details of HCAP, but there's this nice paper out a few years ago where they described this initial HCAP model and the funding for it, called A Case for HIV Cure, and how to get there. In fact, some folks on today's call may have been authors on this, but I will say that much more needs to be done around capacity building in resource limited settings. With REACH, this Martin Delaney Collaboratory, we're one of 10. I'm the program manager for this. We're actively committed to capacity building on the research side of things, and happy to talk more about that. We've actually provided the first in-region digital droplet PCR machine that allows for performing the IPDA assay in Uganda. This is at Rakai Health Sciences Program in Rakai, Uganda. So we're really excited about this potential. They also have the first in-region cell sorter, which was partially funded by REACH. So we're really committed to capacity building, but much more needs to be done on this front. Okay, so when talking about cure-related research in diverse populations, there's this Notice of Special Interest, or NOCI, out by the NIH. So what is a NOCI? This is where the NIH expresses interest in funding specific areas of research. And the purpose of this particular Notice of Special Interest is to highlight the NIH's interest in HIV cure-related research in diverse cohorts of people living with HIV to determine similarities and differences in the establishments, as well as dynamics of persistent non-subtype B HIV reservoirs, as well as in post-treatment control of viremia in people living with non-subtype B. So I'm not going to read through all of this, but in regards to the research objectives, they note that the proposed study should include a particular emphasis on the impact of viral subtype, sex, age, co-infections and comorbidities, as well as geography. So this is really an understudied but important area of research. So talking about co-infections and comorbidities in cure research, the question really is, what is the impact of common co-infections and or comorbidities on the HIV reservoir and potentially HIV cure strategies? So common co-infections could include cytomegalovirus, CMV, Epstein-Barr virus, EBV, hepatitis C virus or hepatitis B virus, herpes simplex virus, tuberculosis, others such as malaria. What is the impact of these co-infections on the reservoir biology and particularly and potentially on cure strategies? And then comorbidities. We know that people living with HIV, even in the era of suppressive antiretroviral therapy, are at a heightened risk of experiencing specific comorbidities. This is driven in large part due to inflammation and innate immune activation. I'm going to talk about that a little bit later in the context of the timing of therapy initiation, but these comorbidities could include cardiovascular disease, specific cancers, diabetes, dyslipidemia, kidney disease, and others. And I'll just note that there have been very few studies to date on the impact of co-infections and comorbidities on the reservoir. I'm going to highlight this one study, where they looked at tuberculosis co-infection, and they found that people living with HIV and co-infected with tuberculosis had higher HIV DNA levels both before starting therapy and more than 1 year after initiating antiretroviral therapy. And so that shows that co-infections may actually have an impact on the HIV reservoir. Much more remains to be determined. So also when talking about co-infections and comorbidities and cure research, there's this recent concept clearance. This concept clearance means that an advisory council at this particular NIH institute has approved this concept for development, potentially into new funding opportunities. And so they note in this concept clearance that as people age with HIV, they are developing various comorbidities and co-infections that significantly impact their health as well as quality of life. And there is emerging appreciation that these various conditions and co-infections also may impact HIV reservoir biology in ways that may be likely to interact with potential cure strategies. However, most research on HIV reservoirs does not incorporate the impact of inflammation, metabolic perturbations, or other pathophysiological processes associated with prevalent comorbidities or co-infections. So we really need rigorous research to address this problem. It's going to require multidisciplinary teams to address how comorbidities and co-infections that are prevalent in people living with HIV interact with viral reservoirs, and how this impacts or may impact future cure strategies aimed at sustained viral suppression. So I next want to turn to antiretroviral therapy access and cure research, and how access to antiretroviral therapy may impact HIV cure. We know that early therapy initiation from a wide body of evidence from many studies limits the size as well as the genetic diversity of the HIV reservoir. It also limits gut damage and resulting microbial translocation, and may then reduce resulting levels of innate immune activation compared to later therapy initiation. It also limits systemic inflammation and may preserve immune functioning and prevent development of immune exhaustion phenotypes. So what is early ART? The definition varies. It's generally within the first few weeks after acquiring HIV to the first 3 months after HIV acquisition. I'm going to highlight one study on this in the Merlin cohort. This was a cohort of people living with HIV in Peru, and they found that participants who initiated therapy within 30 days or less after HIV acquisition showed a steeper as well as a more sustained decay in HIV reservoir measures on therapy. So these results and others suggest a long-term benefit of acute or early therapy initiation on reservoir clearance, as well as overall health. So a challenge in regards to antiretroviral therapy access is that current cure-related clinical trials require participants to be on therapy and virally suppressed. So those are current trials. Again, these are phase 1 trials. Phase 2, they have to assess safety. They generally are, I believe, all require viral suppression. So the question then is, if this intervention works in the trial, will it work in people living with HIV who are not virally suppressed? Are the results from this trial generalizable to other people living with HIV? And then also, what's the safety of the intervention in people living with HIV who are not on therapy? But there may be a possible advantage here in that some cure interventions, particularly some that harness the immune system, they may work better. We still need to research this, but they may work better if given at the same time as therapy initiation. And so in that regard, there's actually a NOCI out on opportunities for HIV cure interventions at the time of therapy initiation. So the purpose of this NOCI is to serve as to identify new opportunities for cure strategies during active HIV infection at or near the start of therapy initiation, or as a potential replacement for conventional antiretroviral therapy, with the ultimate goal of achieving sustained antiretroviral free remission. I guess I'll just highlight from here that additional interventions in combination with the start of therapy may potentially accelerate viral decay, limit reservoir seeding, and induce a vaccinal effect that in combination may induce HIV remission. So there are opportunities here to really explore intervening before complete ART-mediated viral suppression. So another challenge related to antiretroviral therapy access and staying on antiretroviral therapy is that trials, as well as eventual curative interventions, will require treatment interruption. So we really need to monitor viral loads very carefully following an analytic treatment interruption in a clinical trial or a treatment interruption in eventual curative interventions to determine if the curative intervention is working and make sure that people do not rebound. And if they do, we need to resume antiretroviral therapy to promote the health of that individual. So an important consideration here is access to viral load testing. And viral load testing is also critical for other reasons, such as monitoring that antiretroviral therapy is working and not failing. And in some areas of the world, people may only have access to viral load testing once a year at most. That's not going to be frequent enough for curative interventions. And so we need to expand the capacity for viral load monitoring and viral load testing. There is encouraging progress on this, such as point of care, at-home testing modalities, but this really needs to be scaled up and expanded when we think about the implementation aspect of HIV curative interventions, which is down the road. So I also want to go over some additional diversity considerations in HIV cure research. This discussion comes from a paper called Considerations for Increasing Racial, Ethnic, Gender, and Sexual Diversity in Cure-Related Research with Analytic Treatment Interruptions. So globally, women and girls represented 53% of all people living with HIV. This was according to the most recent UNAIDS fact sheet. But between 1995 and 2020, women represented only a median of 11.1% of HIV cure clinical trial participants globally. So women were very underrepresented in cure-related clinical studies. In the United States, Black African Americans account for over 40% of new HIV diagnoses each year. This was according to CDC statistics in 2021. And also 40% of all people living with HIV in the US are Black. However, a majority of participants in cure-related studies are white. And also in the US, an estimated 44% of Black transgender women and 26% of Latinx transgender women live with HIV. This was according to CDC statistics in 2020. However, very few studies report on inclusion of gender minorities. Although I will note that this is increasing, and some studies are separately reporting on and analyzing populations of gender minorities included in their studies. So a quote that I liked from this paper were that despite a disproportionate incidence and prevalence among transgender individuals, cisgender women, and people from racial and ethnic minorities all remain underrepresented in HIV clinical research, especially HIV cure research. And this is important because it's important to understand the efficacy, the acceptability, as well as the feasibility of curative interventions in these underrepresented groups, especially as we're starting to test more cure interventions in clinical trials. There was also this systematic review. It's a little bit older. It was published in 2015 on sex, age, race, and intervention type in clinical studies of HIV cure. But they found that the representation of women, older people, and non-whites did not reflect the national or international burdens of HIV infection. And so, here I wanted to highlight, a question on what is the impact of age on curative intervention safety as well as efficacy. This is really going to be a question and some cure-related strategies because people, not just people living with HIV, but people aging in general, experience what's called immunosenescence. This is an alteration of immune function and dysregulation of immune function due to aging that contributes to increased susceptibility of the elderly to infection, and also, again, dysregulated immune responses. There was in the context of HIV, there was a review written on this. It's a bit older now, from Steve Deeks, but a good reference still. And as I mentioned, it may be particularly age may particularly impact immune-based strategies. And so we need to make sure we're conducting trials in people living with HIV across the age range, and we need to understand what strategies are the most effective in which groups of people. I think we're at the hour, so I'll leave with some key takeaways and discussion. How will these environmental factors impact cure strategies? Today, we only discussed viral clade, co-infections and comorbidities, ART access, sex, race, ethnicity, and age, but there are other environmental factors that may affect HIV reservoir biology, as well as may impact cure strategies, such as someone's diet, their microbiome, their HLA type or other genetic differences. I would just emphasize that much remains unknown. There are calls for research in all of these areas. There is some recent progress, but as the number of cure-related clinical trials is increasing, we need to make sure we're making considerations around diversity, and we need to keep this at the forefront of cure research. So with that, we'll do the Q&A.
So, I mean, fantastic, Dr. Ward. This was really great. I think there was so much appreciation about the comments in the chat for everything that you talked about. And in particular, thank you for doing this with a bit under the weather. One of the initial questions is, can you break down a comorbidity and co-infection, and can a co-infection transform into a comorbidity?
Yeah, there are certain co-infections that can increase risk for specific comorbidities. I can't speak to specific data off the top of my head, but a co-infection is simply another infection with usually another viral, but it could also be bacterial pathogen that may influence reservoir biology. I'll give an example. So influenza, I don't think I have the flu right now, but it's very common. And someone who's living with HIV, they may have a CD4 T cell that's harboring the reservoir. They may have an HIV-infected CD4 T cell that is flu-specific. So CD4 T cells they're important immune effectors as well. And so if this CD4 T cell is flu-specific, the person gets infected with the flu, it's possible that the CD4 flu-specific T cell becomes activated. It's trying to respond to the flu pathogen, and then it proliferates. And then this may result in an increase in reservoir size, for instance. And we're trying to understand how very common co-infections like flu, cytomegalovirus, which over 80% of people globally are living with, how reactivation of CMV, how encounters with these very common co-infections, how it influences or affects reservoir biology.
Great. So there was another question that came in. If you can comment on the genetic variant. So it seems like there were people who have African ancestry seem to have a genetic variant that halves their viral set point. And there's some data out of the PopArt study recently from Zambia and South Africa that there's a higher prevalence of controllers. Is it related to the environment? Could it be their epigenetics? And I also would like
we have to tease that out. Yeah, I think it's
quickly what epigenetics mean, just for those on the call who don't know.
Yeah, I think that's a really interesting question. It's difficult to tease out, but we have to tease out what factor is leading to control, either post-treatment control. What are the mechanisms specifically, and is it genetically determined based on HLA type? This is really important in elite control. Or is it potentially driven also, or instead of, by other environmental factors? It requires really careful studies to tease that out. I can't speak to this particular data actually, but can you repeat the second part? Oh, what epigenetics mean? Yeah, so epigenetics are when you're not just thinking about your DNA sequence, when you're not just thinking about your the sequence of the genes that you've inherited, but epigenetics are other modifications, such as DNA methylation, histone acetylation, other modifications that may influence how or which genes are expressed.
So why a gene turns on or when
Right,
a gene turns on.
right.
So then there's a couple of questions that came in, which is sort of how can researchers convince the community about the HIV cure space and why it's important? And what can advocates do to help further this, the HIV cure research?
Jessica, I might let you contribute to this response, but there has to be ongoing. I think the Martin Delaney collaboratories are a good example. I think more can be done, honestly, of community engagement, but there has to be ongoing engagement in every step of the research process, from the basic science research through to translational research and clinical implementation. There has to be engagement of people living with HIV. They have to be actively involved in the full spectrum of the research process. And we as scientists need to do a better job of communicating advances in research to people living with HIV and to the general public, and create more forums like this for dialogue and discussion.
Yeah, I would just echo that. I think it begins with things like these conversations, but certainly making sure that we're using appropriate language. And you got to shout out Adam as well for saying all the right words like acquisition and not just relying on some of the older papers and the way that they present it. But also, I think this idea of making sure we share the NOCs and the new research that's coming out so that the community is aware, and researchers locally, outside of the U.S. and in places where research isn't happening in the United States or in other contexts, can get informed and involved and begin to put in proposals so that we can expand that access.
Yeah.
And to just maybe a few more
And we have
questions.
to push for capacity building in other areas besides the United States and Europe. Yeah.
Absolutely, absolutely. So there's a great question that came in around the climate of viral control and how does this work in the aging population with so many other older adults have so many comorbidities. And you talked a little bit about this, but I think even not thinking so much about eradication, but this
Right.
idea of durable control.
I think it's hard to know without including individuals with comorbidities and who are aging. Over 50% of people living with HIV in the US are over 50. And we have to, most clinical trials have an age cutoff around 70. We have to make sure we're including these populations in sufficient numbers in clinical trials. It's hard to do right now when we're doing first in human studies, phase 1 trials that are geared towards safety, but as trials are increasing the number of people they enroll, we have to make sure that we're including the spectrum of people living with HIV and perform analyses, not just in individual trials, but meta-analyses across trials on what works in what groups of people. And it's going to take a concerted effort, over the next decade to really understand this.
Yeah, and I would just say that I think that there are a number of groups working on trying to expand access and the inclusion of older individuals living with HIV in clinical trials. And a good place to start is to get involved with the Martin Delaney collaboratories, no matter where you live in the world, because they are such a global footprint and a collection of researchers. Again, if you send me an email, I will share my contact detail. We can make sure that we get you connected into a collaboratory that focuses, that works within your region, or just to get you to stay in contact with advocates who are really pushing this work, some of whom were on the call today. So again, this is a really important piece on making sure that it's not just the actual curative intervention when it arrives, but the research is accessible for all the reasons Adam said. And I think just number one question going all the way back is, are there current immunomodulating drugs that are actually being used for curative
Yes.
research, or are they always being borrowed from other disease areas?
Both. So we have been working extensively with a drug called an IL-15 superagonist that has some beneficial properties. It may be a weak latency reversing agent. It may also allow for certain immune effectors to enter those sanctuary sites and lymph nodes that I mentioned. And this drug actually has been used more extensively in cancer-related clinical trials, but it's now being used in a trial which I'm an investigator on called ACTG A5386. This is a clinical trial testing a combination of two broadly neutralizing antibodies with or without this IL-15 superagonist. We also trials have tested immune checkpoint inhibitors. This was also barred from the cancer field, the oncology field. In HIV Cure, the bar for safety it's very high. It has to be very high because antiretroviral therapy is so safe. So, when we're talking about barring concepts or drugs from oncology, sometimes not all of those will be applicable or we'll be able to use them because they may not be as safe as we need them to be. But there are drugs that are being used right now in cure trials, immunomodulatory drugs, including some that have been barred from the oncology field.
Great. And I think there's another question is just how useful was it to look at cells that do not replicate? I think meaning, especially I think thinking about a person living with HIV in different contexts, how valuable is it those cells that are latent to look at, and do they change depending on the clade or the background environment that a person is exposed to?
Okay. So all regardless of subtype and regardless of clade, an HIV reservoir is established. Are there differences in the reservoir by subtype? We still need to better understand that. They're in the context of active HIV replication or active productive infection. We do know that some subtypes of HIV actually have a higher viral cytopathicity, which means that the virus is more detrimental to the infected cell. It causes more death of infected cells than other subtypes. So we do have some knowledge from subtype differences from active or productive HIV infection, but we're still learning in regards to reservoir biology. And I think, an important takeaway from the presentation today is that all of these factors may be interrelated around environment. The clade, comorbidities, a person's diet, their genetic makeup may all be interrelated. It's very hard to tease out what drives what, but we need more studies to better understand this.
Yeah, and I think that that's such an important point. The idea that, you may have a clade C virus, but move to a place that is predominantly clade B that has a different, background environment, different foods, different, sort of exposures to pathogens, and how that impacts just because you have, one type of clade. If pick up and live in a different environment that's predominant another type of clade, how does all of that play together? And I think researchers are trying to figure a lot of that out. You did have a question that's come in that I want to make sure that we amplify, is to talk about slow progressors versus the window period.
So slow progressors are non-progressors?
So I think people who have acquired HIV, but I think you might consider them to be elite controllers or
Okay.
long-term non-progressors.
Yeah, so when we're talking about HIV and disease progression, we can mean different things. When we're talking about disease progression, we can, there's variability in progression to acquired immunodeficiency syndrome. But when we talk about elite controllers, these are just controllers in general. These are people who control HIV without the need for therapy to either low levels, or in the case of elite controllers, to undetectable levels without the need for therapy. And elite control in particular has been associated with specific HLA types. These are molecules on the surface of a cell that are genetically determined and impact what parts of the virus are presented to the immune system. And so elite control has been associated with specific HLA types. It's also been associated with certain characteristics of the immune response to HIV, a very polyfunctional and effective CD8 T cell response is part of elite control. And so part of it may be genetically determined, part of it may be due to how that individual's immune system initially responds to the virus. And there's also a separate group of controllers distinct from elite control who we call post-treatment controllers. So these are individuals who have acquired HIV went on therapy, but then stopped therapy for whatever reason during a treatment interruption study, or they couldn't adhere to therapy, and then go on to control the virus to undetectable or at least low levels for some period of time without the need for therapy. Post-treatment control is usually lost, but some people can control for multiple years. And it looks like the mechanisms, the immune-mediated mechanisms of post-treatment control differ from elite control, may be driven by natural killer cells in addition to CD8 T cells, and also specific subpopulations of T cells called gamma delta T cells. The mechanisms may differ depending on the individual and whether or not it was an intervention-induced post-treatment control. So, I say all this is we still need to tease out mechanisms of post-treatment control and elite control, and we're learning a lot about this. There was really interesting findings now about 2 years ago that in a group of elite controllers studied by a group from the Reagan Institute in Boston, that elite controllers tended to harbor HIV in distinct genetic regions. So that means the integration site of the virus was in distinct genetic regions that were unlikely to be transcriptionally active. So this means that HIV, probably over time by immune-mediated pressures, was pushed into regions, or these infected cells were selected for over time to be into transcriptionally latent regions where the virus is very unlikely to be expressed. And so a selection over time for these integration site features was described in elite controllers. And so a lot of really interesting findings, even on the basic science side around reservoir biology, just in the past few years.
I would just say then, in related to the window period, I think how could a person potentially what is the potential risk? I think this question is asking about acquisition in that time period as well for these individuals of slow progressors or non-progressors in the window of acquisition.
So the risk
there, I believe in, I don't know if this, if Twibze would like to come off mute and just clarify.
So there are
sure
other
I'm
factors
that right.
that can influence acquisition risk, including co-infections, in particular with other STIs also can influence acquisition. Co-infection with other sexually transmitted infections can influence acquisition risk as well. We're learning, I was actually just discussing with Jessica earlier, that the microbiome may also influence HIV acquisition risk and which strains of the virus someone may be susceptible to.
Yeah, so I think that this is a great place to end. I think, you've answered so many fantastic questions, Dr. Ward, and I think sparked so many that we can continue to go out and talk to our communities about. So I just want to thank you so much for sharing your time and your knowledge with us today. And I want to thank the participants for sharing all of this helpful information and questions and engagement on today's webinar.
Thank you, everybody, for joining.
Thank you all. Have a wonderful day.